So this video is going to be about cancer due to genetic changes. So something to keep in mind is that mutations, especially spontaneous mutations, which are mutations that take place randomly for no particular reason, have the potential to alter genes and potentially lead to cancer. So oncogenes are going to be cancer-causing genes. And so most of the time these oncogenes arise because of a mutation in a proto-oncogene. So proto-oncogenes are going to be normal cellular genes that have the potential to become oncogenes if something goes wrong. So we also have tumor suppressor genes, which are going to be genes whose protein products are going to inhibit cell division and therefore prevent abnormal cell growth. So let's look at some ways that a proto-oncogene can become an oncogene. So if a proto-oncogene gets moved within the chromosome, it can be put under new controls and end up changing the amount of a particular gene, which could lead to cancer. We can also have gene amplification, so we can get multiple copies of this gene, so we'll produce this stimulatory protein in excess and end up becoming an oncogene. Or we can have actual mutations within this tumor suppressor gene that'll alter the protein product, and that could also potentially lead to cancer. So let's look at some cell cycle stimulating pathways. So the RAS gene is going to encode the RAS protein, which is a really important protein in signaling pathways. So it's going to relay a signal from a growth factor receptor, which is most of the time an RTK, and through this cascade of protein kinases and eventually stimulate the cell cycle. So we can see this taking place right here. We have our growth factor binding to this RTK, activating the RAS protein, which sends a signal through these protein kinases to activate a transcription factor and stimulate the cell cycle. However, if we have a mutant RAS gene, then it can produce a hyperactive RAS protein that results in increased and excessive cell division, leading to cancer. So the way that would work is in the hyperactive RAS gene, the gene would be turned on and capable of activating this protein kinase cascade even in the absence of a signal from a growth factor. And so that's how this would lead to increased protein expression at all times and then excessive cell division and cancer. So now we'll look at some cell cycle inhibiting pathways. So signaling pathways are going to be affected by DNA damage, and then the cell cycle is typically blocked until that DNA damage can be repaired, that is in a healthy cell at least. So in this box right here, we have our UV light causing damage to our DNA, which activates a sequence of protein kinases to activate a molecule that's produced by the gene P53. So that P53 will then act as a transcription factor and alter transcription to produce a protein that halts the cell cycle, and so the damaged DNA is not replicated and we don't end up with abnormal cell division. However, if we have a mutant P53 gene, which is normally a tumor suppressor gene whose product promotes cell cycle inhibition, if we have a mutant P53 gene, then even when we have DNA damage, the mutant P53, or it could be missing P53, won't activate transcription, so we won't produce this inhibitory protein and the cell cycle will not be inhibited, and so the cells will continue to divide with this damaged DNA, which has the potential to lead to cancer. I hope you found this video really helpful. All images, unless otherwise stated, are from Campbell Biology's 11th edition. Remember that if you are an enrolled Baylor student, we do offer free tutoring on the first floor of the Sid Richardson building. You can schedule a free one-on-one 30-minute appointment, or you can drop in during any of our normal business hours. For more details, visit www.baylor.edu/tutoring.